start with the first pie graph, and then we'll scroll down to the second one, and then lastly, that table that shows that diversity change, because I think that's important. There you go. So, I mean, it's hard to see this, but that is what I'm talking about when it comes to diversity. I mean, you literally have the vast majority of the important organisms present. Now, when he did DNA, did he have the 18S primer to do protozoa, or was this just bacteria and fungi? Or you may not even know.
This is only bacteria. I am not the best at knowing all these terms that I need to know to speak about this, but identifiers. He said that there's not enough fungal identifiers, if I'm remembering correctly, to be able to do very much fungi. So right now he's specifically focusing on bacteria.
Gotcha. Yeah.
He's hoping to start being able to do more fungi here. Our last conversation, he mentioned that. So this is only bacteria.
Gotcha. Yeah, we're working with biomakers and Mr. DNA at this point.
Yeah.
Because Mr. DNA has the 18S primer, which is the protozoa or the eukaryotes. So it's protozoa, nematodes, etc. Whereas biomakers is more of a really high-end white glove group, but they only do bacteria and fungi. So you really need all of these tools to understand what's going on. I mean, interestingly enough, Biomakers is introducing a new product called BE Crop 3.5, which finally they're starting to look at, okay, if we're going to really understand more about plant overall health, growth, everything, we need to look at soil as both biological, and now they've moved into chemistry. And then finally, hopefully within another year or so, they'll bring in texture too, because that's always a very important thing when you're looking at soils, is what is the sand, silt, clay, organic matter relationship, as well as chemistry and biology, where Mr. DNA is just straight up pure biology. So again, in the future, as we move forward with these new methodologies, we'll start to really get into the weeds and the nitty-gritty on understanding some of these more integral, I want to say, biogeochemical actions and reactions. But so yeah, that's why I was interested in seeing that, because in better understanding it. But at the base foundation, if you have an understanding of the bacterial species with the right pipeline, and a pipeline is basically considered a filter where you run the ones and zeros or the raw data through it. And then you can determine, well, I have this percentage of this type of bacteria. That bacteria is known to do these functions in these different circumstances or within these different communities. And then you can start to weave out a more broader picture about what you can expect the results to come from an application of this type of, whether it's vermicompost or compost. So it's kind of, as crazy as the world is and as scary as the world is, it's a really great time as far as science is concerned, because finally the costs of doing these tests are getting to a point where, I don't want to use the word reasonable, but you can do it. It's not like out of the question. It's not hundreds of thousands of dollars. So I'm looking forward to doing a lot more of that work in the next year.
Yeah, that's what Zach's run into, is the cost. It's just prohibitive. So he's luckily gotten in on the university there, that he's able to get samples in when they're running other samples already. So he can just kind of slip his in the back door and get them done less expensive. So
nice.
Yeah.
Is he still actively doing this?
Yeah, I meant to mention that when you got done saying that, is if anybody wants to check out his work, it's at vermicomposting.com. So VERMI microbiome project, vermicomposting.com.
Fantastic. And I'm sure Lil can pull it up and post it when she can.